Now, the strands of collagen are made originally in the endoplasmic reticulum of cells. That's where the synthesis of collagen begins. Within the endoplasmic reticulum, there's a hydroxylation of prolines and lysine side chains that occurs, and this is critical both for the release of the collagen from the cell and also in providing the strength that's necessary for the collagen to support all the different things that we described. Now, the hydroxylation that occurs on these two amino acids is necessary, as I said, for secretion. If something happens at the hydroxylation and prevents it from happening, then the collagen chain will not be released from the cell. The secreted collagen chains form a helix spontaneously on their own as they are being released. And the helices, as I said, cross-link for strength, meaning that they form chemical bonds between each other. Now, this slide shows the amino acid sequence of a portion of one of the chains. This is what we call the primary structure, as we've described earlier in other things about protein. It contains the sequence of amino acids. And as we look at this sequence, we can see some interesting things. There are 3 amino acids that jump out. The first of those being hydroxyproline. Hydroxyproline is shown as the HPR in this diagram. And hydroxyproline is not a natural amino acid. It's formed by the chemical modification of proline, which occurs after the collagen has been made. It's called a post-translational modification. Hydroxyproline doesn't occur in too many proteins. Proline, we also notice, is very abundant within this region of the collagen primary chain. This abundance of proline is particularly unusual for a helical type protein because proline in most proteins forms bends, and bends are not consistent with a helical structure. The third amino acid that's very abundant here is glycine. Now, glycine we see occurs every third residue, and it almost always occurs adjacent to a proline. That turns out to be really important because glycine has a very small side chain, proline has a very bulky side chain. So putting these two together, glycine allows the space for proline to actually be able to be within a helix. Without glycine, collagen would not have the structure that it does. Now, the other one that may not jump out at you at first, but I have an arrow pointing here, is that of lysine. Lysine occurs fairly infrequently within a given collagen chain, but lysine is very important, as we shall see, for making the cross-links that hold the individual chains together. Now, as I said, glycine occurs every third residue. It is usually followed by a proline, or in some cases by a hydroxyproline. And again, this is to allow the space for proline to exist in a helical chain, as seen here. The hydroxylation of protein is critical, not only for the release from the endoplasmic reticulum, as I've mentioned previously, but also for allowing the overall collagen chain to be stable. This hydrogen bonding that happens between individual proline residues is very critical for keeping a collagen stable at the temperature of the human body. Without that hydroxylation, collagen will fall apart. Now, hydroxyproline almost always precedes a glycine, as you can see again in this sequence. In this slide, we can see the hydroxylation of proline to make hydroxyproline. Proline, of course, on the left and hydroxyproline on the right. Proline is originally put into collagen in the form that you see on the left, and the chemical modification I'm getting ready to describe to you produces the hydroxyproline on the right. Making the hydroxyl group onto proline to make hydroxyproline requires molecular oxygen. And the enzyme catalyzing this reaction, as you can see in the name, is a dioxygenase. Now, dioxygenases use molecular oxygen. They're rather unusual enzymes, usually catalyzing fairly complicated reactions. In this reaction, we can see that there's a cofactor of alpha-ketoglutarate that is needed to make this overall reaction occur. One of the oxygens from molecular oxygen is used to produce the hydroxyl on the hydroxyproline. Well, that requires a reduction, and reduction requires electrons. So how does that overall process happen? Well, the electrons that are used to make the hydroxyproline actually come from ferrous iron, the iron +2 that you can see above the reaction up there. The removal of an electron from iron +2 to make iron +3 is what makes the hydroxylation reaction possible. Loss of ferrous iron to make ferric iron, that is from +2 to +3, is essential for the reaction, but the cell only has a limited amount of ferrous iron. If all the ferrous iron gets converted into ferric iron, then the cell's in trouble and would not be able to exist. That means, therefore, that the ferric iron, the +3, has to be converted back to the +2. How does that happen? Well, that turns out to be one of the most interesting and important reactions that I'm going to describe to you here. And that's because the electrons for making the ferric go back to the ferrous comes from vitamin C, ascorbate in the reaction that you can see above. Ascorbate is vitamin C. So we need vitamin C to keep this reaction going. If we don't have enough vitamin C, we will not be able to produce hydroxyproline, and I've already told you that hydroxyproline is necessary for making stable collagen. So this is pretty important. Well, we have a limited amount of ascorbate as well, so what happens to all the ascorbate if we give up all of its electrons and make the other molecule that you can see here, the monodihydroascorbate? Well, it turns out that the electrons for that come from NADPH, and the cell has a pretty abundant supply of NADPH. So because of the combination of all three of these reactions, the cell can make as much hydroxyproline as it needs. The enzyme that I showed you on the last slide was described as a dioxygenase, and that was to emphasize the molecular oxygen that's used in the reaction. However, this dioxygenase goes by a variety of names, and probably a better way to describe it is as a prolyl hydroxylase, as seen here. The prolyl hydroxylase is depicted in the structure on the right. Now, prolyl hydroxylase recognizes a specific sequence that's found within collagen. That sequence is known as X-pro-gly, where X is any amino acid, pro, of course, is proline, and gly is a glycine. We've seen within the collagen sequence that proline and glycine occur fairly frequently. So the enzyme doesn't just randomly put hydroxyl groups onto prolines, but only those that have in the proline in a sequence like as you can see here. That means, therefore, that this hydroxylation reaction occurs in a lot of places within collagen, and that's very important because, again, it gives collagen some stability. Now, this chemical modification that I've just described is the most abundant post-translational modification that occurs anywhere in the body. The enzyme is located in the endoplasmic reticulum because, as I noted, this activity is essential to get the collagen exported out of the cell. Without the hydroxylation, collagen won't be released, and even if it is released, it won't be stable. Now, this enzyme is flexible. It's a little unusual about it as well. It will modify the prolines of other proteins that have the sequence X-pro-gly. Now, this is interesting because one of the proteins that has a fair number of X-pro-glys is the protein known as elastin. Elastin is, as its name would suggest, a protein that gives flexibility to tissues. Another protein that gets hydroxylated by this enzyme is called HIF. HIF stands for hypoxia induction factor, and the hypoxia is a condition where there's limited oxygen available for cells. HIF is activated when there's low oxygen concentration. Well, how does the cell know when there's low oxygen concentration? One of the ways is by the hydroxylation reaction. You saw that hydroxylation requires molecular oxygen. In low molecular oxygen concentrations, no hydroxylation of HIF will occur. When no hydroxylation occurs, HIF is active. On the other hand, when there's abundant oxygen, hydroxylation of HIF causes HIF to be destroyed. So by modifying HIF in this way, under high oxygen conditions or no oxygen conditions, the cell can tell how much oxygen is there and whether HIF is actually needed.